Darron. He's an associate professor and assistant chair of biological sciences at Southern Illinois University at Edwardsville. And he's here with us this evening to talk about plants in space, using gene and protein expression to understand how plants work with microgravity. So, won't you please join me in welcoming Dr. Darron.
I'm going to put my phone here so I can keep track of my time so I don't run on too long. I promise I'm not checking the blue score in the middle of the talk. But if any of you are compelled to do so, I promise I don't hold it against you. Okay, so I'm going to talk about a project we did with NASA a few years ago, basically putting plants in space. But before I start doing that, I want to rewind a little bit and just think about why NASA would want to fund this kind of research. Like, why is NASA, as a government agency, thinking about funding plant research, right? And one of the main things that NASA has these questions that they want to answer. Some of these questions are, what is the role of gravity in shaping life? This is a big one for me. I am, at heart, a plant physiologist. So what I want to understand is how plants respond to gravity and how gravity informs what plants look like on Earth. So a good way for me to do that is to combine my interest with NASA's and thinking about how gravity impacts life overall. They also want to understand how humans can thrive and survive in space. They want to understand how we can have a presence in space long term. And we also want to understand if there's life on other planets. And so to do that, why would NASA care about plants? Right? There's a few reasons NASA would care about plants. Number one, plants make oxygen. We all breathe oxygen, so that's good. Astronauts on long space trips will need oxygen to survive. They also need food, right? We eat plants. And the third one is just for astronaut well-being. It's very lonely in space, right? It's very different. You're eating a lot of freeze-dried meals. So NASA does a lot of things to help astronauts feel more at home. And one of these things is trying to help them grow fresh fruits and vegetables on the International Space Station so they can have these things to eat. And anytime there'll be like long distance travel to Mars or further, they'll probably have to produce some of their own foods. That's why we're doing some of these things. Am I holding the microphone at an appropriate distance from? Can you all hear me okay? Good? Okay. Okay. So what do we want to know from this work? So the project that I worked on, why are we doing this? And the answer is we want to know what aspects of living in space plants find most stressful. And then we want to know what strategies they use to overcome that stress. And if we can figure that out, then maybe we can use some gene editing technology or just some basic genetics and crossing plants to find the best varieties that will be most successful in space. So that's the overall purpose of what we're doing. If you take one thing away from my talk tonight, this is it. Okay? Plants are not rocks. Even though they are boring and slow, and when you look out at the parking lot, they're sitting there just like the rocks. They are not, right? They are living things that respond dynamically to their environment. They just do it so slow that most of you don't have the patience to watch. But they do respond to their environment. So this is a foliage plant. If you turn it on its side, it changes how it grows. This is in response to gravity. It knows the direction that gravity is pulling, and it wants to grow against it. This is not a light thing, this is 100% gravity. So gravity is really important for plants because they use it to inform basically their entire body. Gravity is not the only thing that plants respond to. They'll respond to light conditions, temperature, time of year, all these things. And they change how they grow and develop based on the world that they're in. Very similar to how you respond physically to the world that you're living in. So, we want to know if a plant isn't growing well, how do we know what's wrong with it? Right? How many of you have a garden? Or at least plants in your house, right? A lot of you. If those plants are dying, how do you know what's wrong with them? It's really tough, right? They can't talk to you like, my kids get sick, they tell me what hurt, like my stomach hurts, okay, fine, I give them some medicine to make that better. When my plants get sick, they can't tell me what's wrong. So it's a lot tougher to figure out what's wrong with the plants. So when we sit in space and they're unhappy, how do we know that they're unhappy and how do we know what's wrong with them? So, Audrey from Little Shop of Horrors, she tells you what's wrong with her. Not all plants plants are going to do that, right? So we have to figure out a way to get plants to tell us what's wrong with them when they're in space, even when they can't talk to us. And so the approach is to focus on their cells. So instead of looking at the whole plant, we can look at them on a cell-by-cell level and tell how the organism is going to react to a specific situation based on which genes they use at any given time. Okay? And so now we'll talk about basically what that means. This all revolves around what's called the central dogma of molecular biology. So the idea here is that all organisms, everything that's alive, has DNA. Okay? And DNA is like the instruction book for that living thing. Every one of your cells has DNA. Every one of your cells has the exact same DNA. So the question is, what makes your skin cells different from your muscle cells, different from your liver cells, right? They all have that same DNA. And the answer is that they have the same DNA, which your genes are in your DNA. You just choose to use different genes. Each cell uses a different set of genes to make it unique. So when a cell uses its genes, it undergoes a process called transcription. So this transcription is basically making a copy of the genes that it wants to use. If I had a giant book that had instructions for making an entire city, if I wanted to give one of you instructions to make just a windmill, I could photocopy those. That would be like the RNA. It's a photocopy of one set of instructions. Then, that set of instructions undergoes a process called translation to make proteins. Proteins are the things that do stuff. It's like the windmill, right? It's the thing that's driving the process. Proteins in your cells are what make everything happen. So we can use this information, how this process works, what genes they decide to use, to tell us what plants are basically thinking. Right? So environmental changes are going to lead to changes in transcription. Changes in the production of RNA for specific genes. Many of you out there have gotten a beverage from the bar. In response to that, your body is producing an enzyme or a protein, alcohol dehydrogenase, that's going to break down the alcohol that you're drinking. You are responding to the environment based on, so on a cellular level, you're responding to your environment, right? So that means transcription changes and we make RNA, and then that's going to lead to translation to make more protein. So what we can do as molecular biologists is measure these things. We can measure how much RNA is there and the changing levels of RNA. And then we can measure the changing levels of protein. And so this, on a cellular level, basically tells us what a cell is thinking about. How a cell is responding to the world. So we're going to use this idea to decide what plant cells are thinking in space. And to do this, we use the International Space Station. You can't see it in this on the screen, but right there's a little white dot. That's the International Space Station. It actually looks like this if you zoom in, right? It is a giant research facility that's orbiting the Earth. It's about the size of a football field. So it's really big. It's been contributed to from countries all around the world, and countries all around the world supply the astronauts that live there, work there, perform science experiments year-round for researchers like me. You can actually see the space station if you want to. There is a website called Spot the Station. If you type in your address, it will tell you exactly what time and what day, when you should go out, and where it will be. Usually it's right at twilight. You can see it, so you can do it anytime you want and see the station. But it looks like a star, basically, like moving along really quickly in the sky. But it's cool, you should actually do this. Okay, so this is where the research is going to take place, right? The problem with this kind of research is the results are super cool. They look like this, right? I get a bunch of, so after we did the experiment, we got all these spreadsheets, they have a bunch of gene names that mean nothing to anyone, a bunch of pie charts and heat maps and things and statistics. So I'm not going to talk about any of that stuff because we were all basically at the table before you got to slide in. So what I'm going to talk about for the rest of the time is basically how we set the experiment up and how things have been going since then. Okay, so overall the plan, like this was the experiment that we set up. We took our seeds, so they're super tiny, like the size of grains of sand, and we planted them on small petri dishes. We did this at Kennedy Space Center. We put these seeds on the petri dish, Those petri dishes fly to the International Space Station on the SpaceX rocket. We kept some on Earth to prepare. They germinated the seeds while they were on the ISS. They grew in the dark for only 3 days. They were super tiny petri dish. There wasn't a lot of time, so they just 3 days they grew. Then they got frozen. Then they got put back on the Dragon shuttle, flown home. We extracted the RNA and protein, and then we compared them. And we said, okay, what genes did the plants that are living in space use as the ones on Earth didn't? And that hopefully will tell us what those plant cells were thinking, what they found stressful about space, and what they were doing to try to get past those stresses. All this work was done at Kennedy Space Center in Cape Canaveral. If you've never been there, I suggest you go. If you are taking your kids to Disney, this is about an hour drive away, and it's awesome. So come talk to me afterwards if you ever think you're taking your kids to Disney, and I'll tell you a bunch of cool things you can do that don't involve you. Okay, so while we were there, we basically had to set up the whole experiment. So these are my collaborators. I'll introduce you to them a little bit later. So the idea here is a little bit hard to see. It's like a jar of liquid media, kind of like Jello, that we, it's liquid at first and then it sets up. So we pour it into these petri dishes and then we sprinkle the seeds on top. To make sure this was all working, I had to bring all this stuff with me. I didn't want to make it there because I wanted to test it in advance to make sure it wasn't going to kill the plants. I didn't screw up making it. Because it costs so much to send things to space, high stakes, you don't want to mess up. So I had to bring everything with me, like in this big suitcase that I bought. But I had to check these bags, and I can't think of anything that looks more like a bomb than several bottles of media packed together in a suitcase. So I put a little note in there, and I said, please, this is a bomb, don't, caress me. If you want to throw it away, please talk to me first. This is for experiment. I never got the little card that said they opened my luggage, so I don't know if they just actually don't screen the bags that come through when they say they do, or what happened, but it survived every time, and I did this 5 times, and they never screened it, so we made it there, so we melted the media, we put the seeds on these petri dishes, and put them in these things called PFUs. It stands for Petri Dish Fixation Unit. NASA has an acronym for everything. Everything. So these are PFUs, you can see it fits right in here, it's like a little metal box. And then we put 6 of these metal boxes in this thing called a brick. It stands for Biological Research and Canisters. And these are modular, and there's a place on the International Space Station where they just slot right in. And so whenever you do research with NASA, you have to fit into what they call their hardware. You have to, they have these parameters that you have to fit your experiment into. So we have to use these bricks, so we have to fit our experiment in here. On January 10th, 2015, the experiment launched on the SpaceX Falcon 9 rocket with the Dragon capsule on top, went up to the space station, and docked. The astronauts unpacked the experiment, and when it got unpacked, they took it out of the cooler. Now it's room temperature, and that caused the seeds to start growing. So they shift to room temperature, made the seeds start growing. They sat there in their brick for 3 days, no one touched them or looked at them, and then after that, an astronaut pulled them out and added this chemical called RNA later that's basically like a fixative. It kept them from degrading any of the RNA, and then they popped it in the freezer, sat there for a month, loaded it back with the Dragon, came back home, and then we had to de-integrate it. We had to taking these things apart, and these bricks apart, in the freezing holes. This was done in a cold room, so you can see that person doing it, wearing a sweater. She had like a scarf on, but it was super cold in there. Then they were transported back to my colleague at Ohio University via this FedEx truck. And the best thing about the FedEx truck is that there was one tiny box in the back, and they got this entire truck. I guess they didn't want to worry about losing the experiment or something. They've got big some general mail. So this whole truck had this one box strapped down the back. The sticker, you can't read it, but it says critical space item. So that will give anyone a hint not to lose it. So then after it was here, we took the plants out and ground them up, extracted their protein, extracted their RNA, and then compared them to the ones that were grown on Earth. And we tried to decide, okay, what are plants in space like? So which genes do plants use when they're in space that they don't use as much on Earth? So we saw a lot of genes change. We saw basically 1,000 genes were different in space, and about 200 proteins were different in space. And so the plant we used had about 27,000 genes in all in total genome, so that's a fairly large amount. So plants, they were confused. Right? So we can tell they were confused because they grew weak. The ones on Earth, in the controls, they grew up off the plate, like plants want to do. They grow up. These ones on the plate, they kind of just stuck to the plate and never even bothered to grow up because they didn't have gravity to tell them. They also had weird cell walls. And we can tell this because a lot of the enzymes required for making cell walls had altered levels in the plants. They were also fairly stressed out. They were generally unhappy. We know this because they had activated stress responses. So a lot of the genes that they would turn on in response to a variety of different stresses on Earth all showed up. A specific one, we also saw altered translation regulators. So remember, translation is the process of going from your RNA to your proteins. We saw that this whole process was very weirdly regulated, and that there were more of all the things required for this process being made than normal. So basically the plant said, oh, there's something wrong, I gotta make more protein. There's also altered splicing, which is a thing that affects the RNA, so they basically edited this RNA a little bit differently too. And then the last thing we saw is that they were hypotoxic. So this basically means they weren't getting enough oxygen. So one thing that's hard, like counterintuitive about plants, is that just because they make oxygen during photosynthesis, they also need oxygen for their normal metabolism, just like you. They have the same parts of their cells, their mitochondria, that use the oxygen to break down sugar, just like you do when you breathe. In space, oxygen does not move around. Gases don't move around like they do here. So now when you breathe out, CO2 breathes out and oxygen moves in to fill that space when you take another breath you have. In space, that doesn't really happen. So especially in the plant's roots, they use up all the oxygen and no new oxygen comes in. So they kind of feel like they're flooded. Right? They feel like they're underwater. So they use a lot of genes that you would find in a flooding response. So you see a lot of altered metabolism. So the way they produce energy, they shift to an energy production method that doesn't use oxygen. You also have this, like you go, hold your hands up in the air for 10 minutes, how your arms start to burn. That's because you're burning sugar without enough oxygen to burn it. You're producing what's called lactic acid. So the plants do a similar thing. They also have metabolism to use less oxygen. They also start soaking up these things called reactive oxygen species, which get made during that type of metabolism, and they're really bad and destructive for everything that's going on in the plant. Okay, so with that, taking 20 minutes up, I need to thank the people who helped me do this research. All guys, me, that's my graduate student, Sarah Hutchinson. She was a master's student at SIUE when we did this stuff. This is my collaborator, Sarah Li, from Ohio University. She was one of the driving forces behind this, and that's Chroma, her graduate student. That's also Maryland's picture of all of us in a copy of the International Space Station's Kennedy Space Center. We all crammed in there for the picture. And then there was a whole bunch of people who helped put this all together at Kennedy Space Center. I'm not going to name them all, many of them, but they did a great job helping us do this because we've never done it alone. And then also my lab at SIUE, these are all the master's and undergraduate students who work for me, who help analyze a lot of these data and will be in charge of doing some of the future projects related to this work. Okay, with that, I think my time is up, so if any of you have questions, I would be happy to answer them. And if you don't, I'll just start showing you more stuff. What kind of plants did we grow there? We grew the plant Arabidopsis thaliana. I knew that question was coming. But I had to get up by like 15 to 20 minutes, so I didn't do it anymore. So this plant is Arabidopsis thaliana. It is the most researched plant in the world, even though you've probably never heard of it before. Its common name is sailgrass, and it's basically the fruit fly of the plant. Right? It's this super well-raised plant, and it's really great because has a really tiny genome, which means it doesn't have a lot of DNA, but it has a lot of genes that look the same as soybean genes. So 99% of the genes in this little plant you can find in soybeans. People care about soybeans, tiny little thing, right? Its flowers aren't pretty, you can't eat it, you can't smoke it. So no one really cares about it from an agricultural perspective. But from a research perspective, it's awesome, and I love it. Because it's tiny too, like here's that thing in the picture. So I have this like just a white box that I can grow a bunch of them in, and it's really, I don't have to like, I can grow them year round, there's no field season, I don't care if it rains 10 inches a week like it's doing now, it doesn't matter. So it's very common. And so the seed action, I forgot I brought these. So you can have these around. So there's some seeds and the petri dish. Here then, you can see the seeds are super tiny, they look like grains of sand. I brought two. So the seeds are in the tube, and that's the size petri dish we use that got put into the PDF.
So when you grab one of these, so like over security, also go back to the machine, do the x-rays,
So do the x-rays affect the seeds? No. It's too quick, I think. And so the seeds were actually, they were checked. They're like in the check bags. So I don't actually know what happens in the check bags. I assume they x-ray them. Like I had to check the media too, right? It's not like I can say, oh no, it's cool, I'm NASA, right? No one can buy that. So there's actually, so one of the other problems in space, there's a lot of radiation. So the radiation they're getting in space is way worse than just a quick pass through the X-ray machine. But that's actually a problem for astronauts and planets and other things going in space. There's a lot of space radiation. Other questions? I have nothing to do with the trivia. So they're going to do trivia after you're like done listening to me, and then they're going to come up and have some trivia. So if you really want trivia, don't ask me any more questions, and the trivia will get you soon.
It seems kind of crazy that you get so much information just from 3 days of growth. You think there'd be more information, more changes over a longer period? Is there a plan to do the longer extended period can actually grow?
So he asked, it seems crazy, it's so much information after 3 days. Is there plans to do something longer term? So, yes and no. The problem is, longer term requires a lot more space. And they don't have, so they do have a piece of hardware now they call the veg. I can't remember what the acronym stands for. But we can actually grow these plants here with adulthood in it, if I could get space in there. But it's really competitive. Like now they're using it to grow lettuce, and the astronauts can eat that lettuce now, and they're super excited about that, so I'm going to get to grow my plants in there. We would see widely different. So it's basically like the idea if we looked at a baby versus an adult. You're using a very different set of genes in some ways than the kid sitting at the table, right? So we would see a different snapshot. I'm not sure I'd say that it's more or less informative, it's just a different stage in life. So it would be definitely informative and helpful to do that, but I don't know if they'll consider it helpful and informative enough to bump the lettuce.
how do Taproom differ from plants on Earth and plants in space?
I guess the very first part.
Tropisms are plants on Earth. How do they differ from plants in space?
So he's asking about tropism, which is basically what I showed, like the first slide, the second slide, the thing was curving up. That's called gravitropism, a plant responding to something and growing in a direction. How do they change in space? So for gravity, there is no gravity. So they don't have any gravitropism at all. They will grow, they don't grow into the media, but mostly they grow just like across the surface of the petri dish. And it's because it's wet, so the surface tension just holds them down, and there's no incentive for them to grow up. If you took them and shined a light from one side, they would all curve toward the light, even more than they would on Earth, because on Earth they're basically windowsill that curves toward the sun, it's fighting gravity. Like gravity's saying, hey, grow up. And the light's saying, hey, grow to the side. In space, there's no gravity, so it just goes to the side full on, and you see more phototropism toward the light because there's no gravity doing the same. Is that basically what answer your question? Yes.
Sorry, his head's not full of rocks, so clearly the computer when his head is not.
Other questions? Go ahead, time to show them else up here video. Okay, I make every student in all my classes watch this video. Here we are. Okay, so this is a sunflower that's growing outside, right? And so north is up. And you know sunflowers follow the sun, right? So the sunflower, the sun comes up, it comes up over here. And now watch, it's following the sun as the day goes along. So this is a 24-hour time activity. So it's following the sun as it passes in the sun's sky, and then the sun goes down, and all its leaves droop. Now watch what it does. This plant knows the sun's coming up over here before the sun comes up over there. Right? And it knows which way the sun is coming from in the morning before the sun comes up. I have no idea how it knows that, but I think that is the coolest thing I have ever seen. And it's one of my favorite things. I pretty much every class I've ever taught, I make the students watch this because this is why I study plants. Right? I know I go through things like plants like rocks, whatever I joke, but this is why I think plants are super cool because they can do stuff like this, and no one knows. If any of you have sunflowers, they're doing this. You're just not watching them do it. So that's easy to know, right? It's so dark. It knows where the sun's coming from now. I wouldn't know if you like dropped me in the forest. I'd never go do this. Okay. Any other questions now that I got to show you my favorite video? No. What do you think? Did you guys like work for NASA? I do not work for NASA. Although once, the closest I've ever come to working for NASA is once at my kid's birthday party, one of the other moms asked me about an astronaut. I'm not. So other than that, no, I don't work for NASA. I am a professor at SIU. But I got to go work with people who do work for NASA, and it was super cool.
Do you have any other experience planned with the space station?
Do I have other experience planned with the space station? So to get those, I have to write grants and have NASA agree to give me money. We made one proposal that they didn't like. One of the weird things that we found in our research is that some of the genes that are in charge of mediating some light responses, like the thing in charge of making them grow toward the sun, basically, that protein, was turned on in space even though they were growing in the dark, which is really kind of weird. We can't figure out why. So we did put in a proposal trying to suss that out. NASA didn't like that proposal very much. We're kind of going back to the drawing board and figuring out that would be that's my next class idea to do this. I would also like to do some epigenetic stuff. I don't know if you're familiar with epigenetics. So it's basically DNA modifications. Like, DNA is modified in ways that are actually heritable, which is kind of the newish last 15-year kind of idea. So I'd like to see what space does to epigenetics as well. But for that, I would need that veggie thing to grow through those and catch to the seeds. Anything else? Okay, I think we're back to Luesse.
Right. So, today we'll be talking about three things. Give you a little bit of background on aerosols and what they are and what they mean. aerosols, and also talking about the process of competing for the Earth Space Air Prize. So starting with the background, what is an aerosol? When you first think of aerosols, you probably think about the spray cans, right? But in reality, it's actually a scientific term. Aerosols are liquid or solid particles that are suspended in air or gas. This origin, it has a Greek origin. Aero means air, sol means solution. This term was actually coined during World War I. And so, if you think about aerosols, you can have aerosols over a lot of different sizes. So, if you think about human hair, that's typically 50 to 70 microns. A rain droplet is typically a couple millimeters. But if you go down to the really small particles, they can be as small as a couple nanometers. Some of the relevant sizes you might be hearing are PM2.5. So that's talking about particulate matter that's below 2.5 microns, and that's kind of what the EPA uses for some standards and regulations as well. And so, in order to characterize aerosols, there are a couple of different things you want to look at. The first one being concentration. So if you think about concentration, it's basically, we're breathing in aerosols right now as we speak, but probably right now it's less, a couple of micrograms per cubic meter. So that means is that if you were to take 1 square meter, or 1 cubic meter of space, you were to suck out all of that air and collect everything, all the particles onto a filter, you would get a couple of micrograms if you were to measure the mass on that filter. So it's a mass concentration. But there are other ways to characterize it as well. So there are number counts, surface area, and so the first way to characterize aerosols is through concentration. And the reason why this matters, here you can see 2 different environments, the same place on 2 different days, very different aerosol concentrations. Obviously, higher concentration aerosols, what will happen is light will scatter, so you actually won't have as much visibility. So this is one of the direct relationships with concentration and how it can affect you on a daily basis. Next one is size. I already alluded to size a little bit, but essentially it's just the diameter of the particle. There are other ways to characterize size, but I won't really get into it. But generally in the environment, you'll see something called a size distribution. So you'll never just have one single particle size, you'll typically have a distribution. So that distribution can range anywhere from a couple of nanometers to a couple of microns, and it just depends on what you're interested in measure. So some example aerosols that you'll see in their sizes. Cigarette smoke is typically less than 100 nanometers. If you look at emissions from power plants, it can range anywhere from 300 nanometers up to 10 microns. If you have an inhaler, those are typically a couple of microns, and those are designed so that they can penetrate deep into the lungs. Or when you sneeze, you can have really small particles that are a couple of microns, or even really big droplets that are on the order of a millimeter. And the last one, this is the hardest thing to characterize as composition, and partly why we're doing what we're doing. Because you're going to have aerosols, but aside from knowing the size and concentration, what are you looking at? Obviously, different types of aerosols can have different effects on your health. So, for example, cigarette smoke are typically volatile organics, whereas you can have inorganic aerosols and they have very different implications and properties. Just some recent examples of why aerosols matter. The California wildfires were a huge issue. These smoke lingered a couple of days, and even though the wildfires were happening in certain areas, because of atmospheric weather patterns, different communities would get affected and get exposed at different types of time frames. So being able to characterize exposure to wildfire smoke is actually becoming a big issue. This one actually is a picture of geoengineering where they're trying to actually spray particles into the atmosphere. As you know, climate change is, people are trying to figure out ways to address climate change. So what some scientists are actually proposing is spraying particles into the air so they can become seeds to form clouds. So this is more of a drastic measure, but actually, large-scale geoengineering is actually being discussed and explored. So on to the second part. Why does NASA care? I already talked a little bit about it, but one is NASA has a lot of satellites floating around, and they have all these instruments that are actually taking images and taking measurements of the atmosphere. So scientists have some really amazing, NASA has amazing scientists that are doing a lot of work on modeling climate change and different atmospheres. But also, in the space station, astronauts are actually getting exposed to nanoparticles. So, some couple facts about the space station. It's about a 900 cubic meter habitable volume, about 6 bedroom house. It's been inhabited for 18 years. 6 crew members at a time since 2009. But the most interesting thing is that it's the same volume of air all these years. So typically if you're in a room and it gets stuffy or something smells weird, you can either just leave the room or you can just open a window. Or you can trust that ventilation will eventually clear it out. Well, you can't really do that in space because there's not really an atmosphere outside. Everything is a completely closed system. So everything you bring in has to be treated or taken care of. There's really not a good way or effective way to just vent everything out. So from what I've heard, it actually does not smell very good when you first get up there, but you kind of get used to it. And another thing, what happens if there's a fire on the space station? So actually trying to find ways to research sensors that can actually detect fire. The most interesting thing is that fire burns very differently in space. You can see the flame that's typically on Earth, and here in a microgravity environment, the flame has a very different shape. And even the way that smoke disperses will be very different in a microgravity environment. So one of the researchers that I worked with at NASA did a study where she actually sampled particles in the International Space Station. Initially, the thought process was people didn't think that there were nanoparticles in space. So they actually flew up some samplers into space, and they found a lot of interesting things. So here I'm just showing some pictures of particles. So what she did was she actually took these filter samples, brought them back, and analyzed them using something called scanning electron microscopy, which is basically being able to really zoom in on particles that you can't see with visible light. So one thing is TiO2 nanoparticles. The source of this was that astronauts actually have to wear sunscreen when they're near windows because of the high levels of radiation. And because they have to wear sunscreen, you can actually see that there's sunscreen that's being picked up from these filter samplers. So they're wearing sunscreen, but they're also kind of breathing TiO2. So not the best situation. Other things that they were finding were cubic salt particles, because scientists sweat even when they're in space. You would also find this especially in the eating area. So here you can see the eating area where all of the different bottles of sauce that they had, including sriracha and barbecue sauce, all had Velcro on them so they wouldn't float around. But this is where they found high concentration of salt particles as well. So this is the third part, where I'm going to be talking a little bit about the process for competing for the Earth Space Air Prize. So NASA actually partnered with the Robert Wood Johnson Foundation on this. This is the first type of competition that's happened. They were actually deciding whether or not they wanted to do a competition on air quality or water quality. Luckily for us, they picked air quality. But really what they were trying to do is catalyze the development of new technologies around aerosol sensors because they realized there was a huge need for better sensing technologies that could be deployed in space, but also on the Earth. So the team includes Dr. Biswas. He is the chair of the Energy, Environmental, and Chemical Engineering at Washington University in St. Louis. He was also my professor, so I did my Ph.D. at WashU in aerosols and air quality. I studied combustion synthesis, so actually I got to burn stuff for my Ph.D., which was really fun. I also had worked with Jiayu Li. She's a current Ph.D. student at WashU. And then Tandeep is my co-founder. So we both did our PhD together in the same lab and decided to start a company after we graduated instead of getting real jobs. And this is a picture of all of my past professor students. So he recently had a reunion where a lot of his former students, came to the International Aerosol Conference. My professor's kind of at the peak of his career. He just won a bunch of awards and won the National Academy. But A lot of his old students are actually professors now. Some of them are in industry, but he's really made a huge impact in the aerosol community. So we had a really nice get together about a year ago. So you can see I'm like right back there, Tandeep's there, and Jiayu's in the front, and the professors in the middle. So the timeline of the competition. So we had to submit a proposal for a new technology in January, and what happened is that they would review the proposal, and then once you got informed, they would pick 3 finalists. 3 finalists that would then have to build a prototype in 6 months, and then take that prototype to NASA. We went to the Glenn Research Center. That's where a lot of the aerosol characterization equipment is located at. So there were about 20 teams, I think, and with the 20 teams, they picked 3 finalists. We had to write a detailed long proposal for this. And what we're essentially proposing is a multi-wavelength aerosol spectrometer. I won't go into too much details about the technology, but essentially what we're doing is we're trying to shine light on particles and figure out how we can interpret the data from that light using lasers and the photodetectors to determine what is the concentration, particle size, and also the composition of the particles. Typically, right now, with a lot of the current sensors, you can only measure size and concentration. It's very difficult to measure composition, so we're trying to find ways to do that in real time in a scalable manner. And so once we submitted our application, there were judges that were selected by the NASA scientists. These are all professors and experts in the field of aerosol science. They got to review all the applications and pick the top 3. And here are all the finalists. I think we're all pretty happy because we were just happy to be able to go to NASA and test out our devices. But the first one is John Wilkins. He's a professor at Colorado State University. And here are his postdocs and his PhD students right there. And then David Woolsey with AirSpec is another great startup in the Bay Area that developed their own sensing technology. So all these guys were PhDs in aerosols in the small community, so we all got along really well. And then we had more judges. So these were the judges where we actually had to show our device to. They were looking at our data. So the way the competition would work was that we had 2 days to test our sensors. We would all test it in the exact same environment. At the end of the day, we had to send our data to the NASA scientists where they would get to take a look at it. And then a month later, they would tell us who the winner was. And the system that we tested in was called the Gases and Aerosols for Spoulding Polymers Laboratory. So basically what they did was they built a chamber where they could burn stuff and then measure what came out of the aerosols that came out of burning the fuel sources. So this picture's not very clear, but essentially you had a furnace and then you had a bunch of instruments that would measure the particles in the chamber. This is just us kind of at the Glenn Research Center on the first day. They let us take a look at the chamber to see, how we would set it up. So we would all basically put our sensors down here, and then they would basically feed in all of the aerosols during the experiment, and it would be vented out here. So this chamber was originally built to do a lot of experiments to test fire and combustion and different types of fuels and materials that could potentially burn in the space station. A lot of the issues are the fact that the space station has a lot of electronics. So what happens is if electronics overheat, they actually emit a lot of particles. It's through a process called pyrolysis. And these particles are really small. So NASA's trying to find ways to detect these particles and also be able to figure out, are we detecting particles or are we detecting smoke? This is a huge issue in the space station that they're trying to figure out. So that's why they built this chamber. We compared our sensors with a couple of reference instruments. And these are all very expensive, $20,000 to $100,000 instruments that give you size and concentration. And that's how they would know whose sensors was the best, is that we would send our data, they would look at our data compared to these data from these instruments and decide who was the winner. So this is just me trying to get my sensor to work, and then my competitor looking over my shoulder to see what's going on. And then what we did was we tested 12 different aerosols. I won't name all of them, but we had everything from things like foam to reference aerosols to cigarette smoke to diesel smoke. So we just did a whole bunch of different types of aerosols to test their characteristics and see how our sensors would perform under different conditions. So the NASA scientists came up with a really scientific way to generate diesel. What they would do is, there was a parking lot outside, someone would drive up, turn their car on, they would take a trash bag, and then let it fill up with diesel, then run into the chamber, and then just kind of pump the bag and feed it to the chamber. So that was just kind of an interesting experience to see what's the most cost-effective way to generate diesel fumes. Oftentimes the best solutions are the simplest. So here are just some test results. We tested against lunar simulant. So at the Glenn Research Center, they actually were trying to develop tires that would be able to, that they would use on the moon. So they had lunar dust simulant. So they took that same lunar dust simulant, aerosolized it, and then we would see how essentially perform. So you have a couple different size distributions, anywhere from 300 nanometers up to 4 microns. We also tested actual aerosol spray cans. So that first picture I showed you, most of this is actually fairly auto-dispersed for aerosols, anywhere from 1.3 to 4 micrometers, which is pretty standard for aerosolized droplets. And then we also tested diesel particulate matter. So this is why size and concentration are very important in understanding, is that you can see here the concentration is really low, 2 micrograms per cubic meter. Just for reference, EPA standard is about 15 micrograms. In India right now, they're probably reading about 100 to 400 micrograms. So, in terms of mass concentration, it's pretty low. But if you look at particle counts, it's actually really high. So you have to really be careful when you're talking about exposures and thermal concentrations. You need to both understand the particle size and the concentration. It's very important. And then, luckily, I didn't find out during the competition, but we ended up winning. So, thank you. I felt like I had to take the picture of the cake that I had before and after, but the cake was for everyone. But it was just a really great event because, the type of work that we did usually takes, a couple of years, a lot of funding to execute on. But with me, Jiayu Biswas and Tandeep, we were on a tight timeline and didn't have a lot of resources, so we were able to develop a new technology in 6 months, which was really exciting for us. So the next steps is that we're going to do some more testing to improve the technology that we developed, we're talking to the NASA scientists to figure out what steps we need to take to make this ready to take up to space. So there's a whole process with that to make it spaceflight ready and compatible. And then we're also really excited to explore applications on Earth as well. So NASA has a lot of initiatives with environmental air quality sensing. So we're talking to NASA about air quality as well for smart city applications as well. And on the side of that, we're also working with environmental protection agency, U.S. State Department, and National Institute of Occupational Safety and Health on our sensing technologies. And so for just the company Applied Particle Technology, we work a lot on the NASA technology, but as a company, what we're actually trying to do is work on industrial air quality. So one thing you might not know is OSHA estimates there are about 50 to 70,000 workers that die a year due to airborne pollution exposures. So this is in industries like oil and gas, mining, construction. So we thought this was a huge issue where we could really make a big impact. So what we're trying to do is basically develop wearable exposure monitors. And so miniaturize these sensors into a wearable so that when people go to work, they can actually see what they're being exposed to in real time. And they're exploring a lot of really great applications for this. So some things include deploying networks of sensors so you can get your air quality concentration in real time. Also, we're working with researchers at UC Riverside. They actually took a couple of our wearables and put them on kids in Southern California, and they were actually able to identify where their exposures were occurring throughout the day. Interestingly enough, retail environments had the highest exposures for some reason. And the professor I talked to, her hypothesis was that they were vaping. So vaping was causing all the exposures. So, yeah, so that's it. That's my company, Applied Particle Technology. Have any questions or feel free to contact me or just come and ask questions. Our website's appliedparticletechnology.com. Thank you to NASA, thank you to the Robert Johnson Foundation, and thank you to WashU. This was a really exciting experience. Thanks. Any questions?
How do the aerosols behave differently in space, in microgravity? Do they behave differently? Is it hard to measure? Is it something you have to build a different algorithm for?
Yeah, so like the fundamental processes, the main thing is just gravity. So if you get to the really small aerosols, the primary process is still diffusion. If you get to large aerosols, then gravity will play a bigger effect. But the equations are all pretty much figured out. So there haven't been too many experiments, but also it's because the real main difference would be gravity, and it hasn't really been studied too much. So really, it's not probably would be too big of a difference. Yeah.
So your equipment would basically identify the aerosols in space, right? How do they get those out of the International Space Station after they're there?
I should have put that slide up. So they actually have these filter racks in the space station. So one of the issues is they have to fly up activated carbon and have the filters, and then they have to fly them back down. One thing I found out was that they charge $24,000 for each filter. Basically the same filter that you probably use in your furnace. So one of the NASA scientists is actually trying to figure out, do we really need to be flying those up every single time, trying to figure out how can we test these to make sure they're actually working. But what actually the astronauts do is, once the filters are clogged up, they'll actually take a vacuum cleaner in the space station to clear out the filters. So what ends up happening with filters is that as the dust collects onto the filter, they clog up and they don't work as well anymore because you can't get air flowing through the filters, so they stop working as well. All right, thank you.
Well, thank you very much, Dr. Fang and Dr. Luesse, for outstanding presentations tonight. I'm Chris Hefner, and John Mackey and I are solar system ambassadors with NASA and the Jet Propulsion Lab, and we're excited to bring some of the local science that relates to space explorations to all of you. I was reminded with Dr. Fang's presentation about this has been a problem with space exploration from the very beginning. In the Apollo 8 mission, Jim Lovell actually accidentally triggered his life vest during the early part of the launch. The life vest was for splashdown at the end and was a CO2 cartridge. So here he had this life vest that was filled with carbon dioxide, and they didn't want to just let it into the capsule because that would increase the concentration of carbon dioxide. So they had to use the vacuum cleaner that was on the capsule to be able to vent out this carbon dioxide. That goes all the way back to December of 1968. So thank you very much, all of you, for coming out here during Game 5 of the Outstanding Finals. We'll see if we can do another event maybe in early November. We'll schedule it for the 7th game of the World Series, just in case the partners are there. And so thank you very much. Thank you to Natalie Blackman and Rose Jansen from the Academy of Sciences St. Louis for partnering with us and bringing things over here. Thank you to Julia and the entire staff here at 4204. And hopefully we'll be able to do a lot more of these for the Metro East area. And thanks again, everyone.